Gentlemen, welcome to segment 3 of our conference on research and innovation in seed development. So today I'm your moderator for this session, and it's my pleasure to guide you through a series of insightful presentations by esteemed experts in the field. For each presentation, I will invite our speakers to the front to take their designated in front before they begin. So without further ado, let us welcome our first speaker, Dr. Setival Poraya Gonda. He's from GWG, or we call Green World Genetics Sdn Bhd at Batu Arang, Selangor. Please come, Doctor. Come. Okay, for the second speaker, we have Mr. Tan Chung Chuang, the Chief Executive Officer, CEO, AGRO Primajaya Sdn Bhd. Please come, Mr. Tan. Okay, maybe he will, yeah, outside. Okay, never mind. So we go to the second speaker. We have Mr. T. Nagaran K. Haridas, Solution Manager from SIGPA. Mr. Tina, please come. And the fourth speaker is from APEC, Mr. Mark Monaghan. Please come, Senior Product Specialist. Okay, without further ado, I would like to introduce our first speaker, Dr. Setival, as a senior breeder in Green World Genetics. His expertise is in plant breeding. The academic background, and currently he's just get the PhD in plant breeding. And for the masters, he has a genetic and plant breeding. And for the, he actually 24 years of commercial plant breeding experience with Syngenta, S.E.E.D.S, and Green World Genetics in China, India, and Malaysia. So many countries that you have been working, yeah, Doctor? Okay, without further ado, I will call Dr. Setival to come to give our presentation today. Please.
Good morning to everybody. Today, I am going to present the cucumber breeding for high yielding and Begoma virus resistance. The standard practices what we are doing in the commercial plant breeding sectors. Just I would like to explain some key methodology and the results that we obtain from the Green World Genetics. Cucumber is a very important vegetable crop. It belongs to the Cucurbitaceae family, the third most important economic vegetable crop in the world. But is cultivated around 2 million hectares with a total production of more than 93 million metric tons in 2024. The top 5 cucumber producing countries in the world is China, Turkey, Russia, Ukraine, and Mexico. The China alone accounts for more than 80% of the global cucumber production, and also 59.5% of the global cucumber area. But the Netherlands ranks the highest in productivity. The productivity in Netherlands is 638 tons per hectare. Around more than 18 countries, their productivity ranking above 100 tons per hectare. But still, Malaysian productivity is less than 20 tons. Even though China is 58 tons per hectare, but Malaysian cucumber productivity is just less than 20 tons per hectare. So in Southeast Asia, even though Malaysia is highest in productivity, but still when compared to other countries, like in European countries, is very far low. Next. So when you look at the problems in cucumber productivity, why we are unable to achieve the highest productivity, the first problem is the scarcity of good quality seeds. When you compare the cucumber seeds in European countries and other multinational companies, the seeds available in Malaysia is not comparable to that standard. And the biotic factors is also a very important problem, and adverse climatic factors, because in Asia mostly it is an open field cultivation compared to the greenhouse cultivation in Europe. So this is also another challenge for us. Then high perishability of the cucumber fruits, lack of investment capital, and inexperienced and unskilled laborers, these are the main concerns. So specifically, if you look at the way how we can improve the productivity in open field cucumber in Asia, the first one is we have to improve high yield by bringing the resistant to biotic stress, especially the resistant to Begoma viruses. Next. So Begoma viruses are getting very serious problem in Asia. As you know, the virus, there is no control measures and there is no chemical, even though we can control and manage viruses through vector control, but this is just a temporary solution. We cannot permanently control any viruses. So there are 24 Begoma viruses totally reported in cocoa beets worldwide. But the most common Begoma viruses is only the listed 4 the tomato leaf called New Delhi virus, squash leaf called China virus, squash leaf called Philippines virus, and tomato leaf called Palampur virus. These 4 are very potential and very strategic viruses for Asian growers. In management, it's very difficult because Begoma viruses are very systemic. It's like all other viruses, these are very systemic. And then they're very more diverse and they constantly evolve. And because of the different mutation and recombination events, is very constantly evolving into different strains. This makes it very difficult to bring in a resistant gene that can efficiently bring the resistant level in cucumber. And moreover, the whitefly carries a multiple virus because whitefly is the vector, insect vector for this one, and they carry a multiple viruses. So even one bite, the whitefly will bring a billions of virus particles. They can accumulate and then transmit to the another plant. The only solution is we bring seeds and seedlings in a very protective environment. This is one of the best solution. And we can control vectors, but even though it's not a permanent solution, mostly right now, if you don't have the resistant variety, the growers have only one option, the controlling vectors right now. Until the plants get infected by the vectors, then no problem. Once it is infected, nothing can control. So the using resistant varieties are the permanent solution. Now we can explain later. Yeah. So next. Yeah. There are 2 ways of bringing resistance in cucumber. One is the conventional breeding, and another one is the genetic engineering approach. But for Begoma viruses, bringing for a conventional breeding is a very long process. It takes multiple years to bring the resistant level. And then, fortunately, we have QTLs already reported, even though it is controlled by a major recessive gene. But we have identified QTLs also for these Begoma viruses. Then 4 subclasses of gene family also identified for this virus. By genetic engineering, RNA technology is also a possible one. Different scientists already tried this method. This one is beyond the cross border. So we don't need to worry about the species restriction and barrier actually. But it needs a very complex regulatory approval, even though it is very convenient than the GMO, because the gene editing is getting a flexibility in many countries now, including Asia. Asia and Europe are very strict for genetic modification, but they are now liberal now. But when it comes to the gene editing technology, many countries are opening an avenue now. So last month also, India also approved a gene-edited paddy, so for the commercial cultivation. So that gives a very clear signal that the gene editing can be a potential tool in future for plant improvement. Next. So these are all the symptoms in cucumber. You can see the last, the D is a healthy plant. But if you see A, B, and C, you can see the curling of the leaves, chlorosis, and the shortened internode. So the plants will get stunted, and there won't be any productive growth afterwards. Next. So to identify the virus, Bergamo virus, PCR is a very reliable technology. So even though you can use other ELISA or many methods, but the PCR is the final confirmation. So this is the protocol we use to identify the Bergamo viruses in cucumber. Next. So after identifying the virus by PCR, then we can also specifically identify the species, because Bergamo virus is not a one virus. There are multiple viruses in Bergamo viruses family. So you can specifically identify the species by sequencing the Bergamo viruses, what you C3 sample, and then what we collected from our GWG R&D station. And then we amplified by PCR, and then we used this amplified band to again sequence. And we used the Mega 11 software for the gene editing and alignment and assembling. And later we used the BLAST from the NCBI database. And phylogenetic analysis, we used it in the virology, plant virology. Next. So these are all the results in PCR. You can see the primer 1800 and 2300 is a very efficient one, because it can identify the viruses in all the samples we tested. So all the 9 samples, you can see the band. So this primer can be an universal primer in future. Whatever crop, maybe it's a crop, it's not a crop specific, but it's a virus specific. So can be an universal primer. This sample is in the top. That is 100% matching with the tomato leaf curl nudity virus. So also very closely matching with the other all reporter viruses from other scientists also. So from 97 to 96.7% is all closely matching with the other variants of tomato leaf curl nudity virus. Next. And even phylogenetic analysis also, you can see 2 different methods we used, and but in both the methods, you can see sample 3 is very closely related to tomato leaf curl nudity virus. So this confirms the tomato leaf curl nudity virus is a very serious virus in Bergamo virus in Asia, especially cucumber in Malaysia. Next. So once we identify the virus, then we have to think about how to bring the resistance into our hybrids. So for the hybrid breeding, we used 48 breeding lines from the male and 31 breeding lines from the female. And then 2 lines were balancing both male and female, so we kept in the both category. So this is the germplasm what we used, and this by default it must have the resistance to Bergamo viruses, because they are controlled by recessive gene. Next. So for making a top cross hybrid, we use one tester, one mentry from the female group, and we made a top cross with all the male. And in the same way, we used 2978 from the male group and cross with all the females. So this is how we do the plant breeding first test cross. So we call it a top cross method. And based on this hybrid evaluation, and we identify the breeding lines with the high general combining ability. So if the lines having the high GCA, they have a high potential to bring a high yielding hybrid variety in future. So we cannot cross whatever lines we have and then in multiple combination, and then this is a very tedious job, because you will get too many hybrids for evaluation that is not easily manageable by the plant breeders. Next. So based on this one, you can see male line, we identified the 17 high GCA parental lines. And then for female, we identified the 11. So we made all possible combinations. And then you can see from the male group, 17 breeding lines. We only picked up the positive GCA lines with the positive direction and the highly significant GCA lines. So these 17 lines we identified. Next. In the male group, in the same way, after top cross, these 11 breeding lines in the female group also we identified with the high GCA, significant and positive GCA. Next. So later we used this 17 male and 11 female, and we made all possible combinations. And we developed 185 hybrids. But in 185 hybrids is also very very big number, so we cannot evaluate all the numbers in multi-locational trial. So we selected whatever hybrid yielding in female parent more than 25 gram, we selected, and then we carried forward to next multi-location trial. Next. So these are all the hybrids of whose female parental lines yielded more than 25 grams seed per plant. So this is very important because this is a commercial breeding. So if the female parent is less seed yielding, so the seed production is not a commercially successful one. So we cannot make any profit and we cannot get any ROI. So next. In hybrid evaluation is a very critical part because you have to choose the location and also you have to choose the season also. It's a very important one. So all the 20 hybrids we planted in 2 different seasons in Terengganu, and then 20 new hybrids and 26 parental lines, and along with 6 cucumber commercial check hybrids from Southeast Asia. We included total 52 entries in the 2 multi-locational trial. Next. This is a scoring method we use for Bergamo virus. So even though we identify as a Bergamo virus, but still you have to score, phenotypically we have to score. And 0 is a highly resistant and 5 is a highly susceptible. So we score this one in at 65 days after sowing in each hybrid trial. Next. You can see these are all the newly developed hybrids in GWG. The GWG 8, 11, 12, 13, 18, they all finally shortlisted from this trial. And we can see the commercial varieties C5 and C6 and bottom. So only GWG 18 is a white color. The remaining all green color, but is matched for the different markets. Next. So this is the ANOVA table for the trial 1, first season trial. Even though the genotype and replication both are highly significant, if you go for the ETA square, is most of the variation in the trial comes from genotype. 93.4% variation coming from genotype. So the replication gives only 1.1% of the variation. So that is a very negligible. So it indicates that the trial is perfect, and then most of the variation comes from genotypes. Next. In the season 2 also same. Both genotype and replication showing highly significant difference, but the ETA square confirm the most of the genetic variation comes from the genotype only. Next. In the yield per plant, this is a post-hoc analysis after the ANOVA. We did the Tukey post-hoc analysis test because this one is a very reliable test for the post-hoc analysis because it encompasses the pairwise comparison and efficiently reduce the type 1 error in any statistical analysis. So in trial 1, you can see the first group 1 is the GWG1. This is significantly different than all other entries. So in the second group, you can see 12, 4, 13, 8, 7, 18, all are significantly same, but different from the 11. So in trial 2, the first high yielding is in the group 1. So 12, 13, 11, 8, 2, 14, all are high yielding and statistically different from the group 2 and group 3. But we can see the other group entries also here. Next. Later, we did the combined analysis for both the trials. So this is also very important because you should know how the 2 different trials are interacting, the seasons are interacting. And we can see the season and genotype also highly significant difference, we got it. But still the ETA square is 53% come from genotype and 31% come from season. So the season and genotype is only 9%, is very negligible. But it's also indicating a rank changing interaction. That means the order in the performance is different from both the trial. That indicates the genotype season and genotype significant difference. And you see the Tukey post-hoc analysis test. Again, the same entries are performing. This is confirming with the trial 1 and trial 2 individual trial analysis. GWG 11, 12, 13, 8, and 18. These are all the selected hybrids, also performing in the combined ANOVA. Next. In the Bergamo viruses scoring also, you can confirm all the selected entries, they have a very high level of score of 1. So the high yielding with the high Bergamo virus resistant, we already confirmed in our trial. So next. This is a correlation. It's also a very good indication how the traits are correlating in cucumber. If you look at the trial 1 and trial 2, the number of fruits and yield is also correlating, strongly correlating. So that means the more number of fruits is ultimately giving more number of more high yielding. And also the fruit weight and fruit length also highly significant correlation. But one thing is very surprising for us. The Bergamo virus, even though it's very important, but it's not strongly correlating with the yield. This is indicated in both the trial. So this indicates the two traits are independent. So if you bring resistant for Bergamo virus, doesn't mean that your hybrids are giving high yield, but the resistant level for Bergamo virus can secure your yield when there is a disease pressure. But if you have the resistant gene, that will secure your potential yield. But in case if you don't have the resistant gene, if there is a disease epidemic, you will lose completely all the yield potential of the hybrid. That's what I want to emphasize here. Okay, next. Next, yeah. So as a conclusion, identification virus is, we confirmed the wider distribution of Bergamo viruses. Specifically, this primer, 1800 and 2300, can be an universal primer. And then we identified tomato leaf curl noodle virus in the trial. In hybrid development, we confirmed the top cross method and the GCA estimation is a reliable technology in breeding tools to identify a potential hybrid. And then seed producibility also we can filter. And in the beginning itself, I highly recommend all the breeders to filter the seed producibility. So in future, when you commercialize, you won't get any surprise. And then hybrid evaluation, we identified these 5 hybrids as a high yielding one. And except the 18, all are green color, 18 is a white color one. And all the hybrids are already commercialized by GWG. And 4 and 7 are only recommended for season 1 because they specifically perform in season 1, not in season 2. And there are 2 mega environments because season 1 and season difference. And the Bergamo virus resistant level in Czech varieties are ranging from resistant to, our hybrid ranging from resistant to medium resistant, but in the Czech variety C5 is a very confusing result because medium resistant in trial 1, but susceptible in trial 2. That means it's not a reliable one, yeah. That's all from my presentation, yeah, next, yeah. If you have any questions, you can ask me, yeah.
Thank you, Dr. Peksaktival. Okay. Next, I would like to call
okay, that's all.
The Q&A after
okay.
all.
Okay, thank you. Thank you so much.
Okay, Mr. Tan. Tan Chung Chuang is the Chief Executive Officer from Arus Prima Jaya Sremberhad. He was work in tissue culture from since 2000, and today he will present on in vitro propagation for the commercial production of elite planting materials, focusing on progress, potential, and challenge. Okay, now I present Mr. Tan.
Okay, thank you very much. Hello. Good morning to everybody. Okay. I from the company is Arus Prima Jaya. Then I'm the CEO of the company. Okay, today I present is in vitro propagation for the commercial production for elite plant material progress, potential, and challenges. Okay, actually I involved in the tissue culture is since 1998, and then the largest scale tissue culture planting production in the banana of the Malaysia type about 13 variety, something like Brangan, Skaki, and so on. Then also the ginger variety. Then the pineapple is mostly is MD2. Okay, next. Okay, today is an introduction for the define in the vento propagation, review to agriculture and horticulture, importing for the elite planting material how to improve to the high yield, disease-free, and a uniform for the yield and the planting material. Then the principle for in the vector propagation, but so on we know for the tissue culture, we have the 4 steps for this basic for the production. The mean is for the micropropagation step, the inventation is sterilization, and then afterwards we need to do selected for the mother plant is free for disease and free for virus, and then only can take on and continuous in the lab to do the multi-propagation, and then the second point is a multiplication. The mean is the mother stock. How many times we can do the duplicate, or the time for this mother stock can stabilize, no become the forming for the growing. Then the third is rooting, and then the number 4 is accumulation. Okay, before we know about for this tissue culture of this planting material, then we look about for the history, the background for the history for plant tissue culture, the milestone and in commercial application, and also the key contributors to the field. Then overview, actually tissue culture in this history is since 1902, and then the father of the tissue culture is Gabriel Heberland. Then he is starting for this develop for the tissue culture at the times, and then the develop for the nutrient and the media and the basic accepting technique, and then success in the culture callus formation and then the organization. And then afterwards, we see for the second progress is a technology breakthrough. It's 1960 up to the 1920. Between these 20 years, becoming for the MS media for this up to now, I think a lot for the lab in the tissue culture still use the MC media. It's very famous for their using. And then the point is first successful for the commercial micropropagation is orchid. Then somatic embryo and then the protoplast also function developed in the times. Then used in the plant breeding and germplasm commission begins. Then when this commercial era be happen and 1919 up to the year 2000, and then this period allow for this rapid growth for the tissue culture global is starting for the mass production. Okay, and the war for the adoption for the banana, sugarcane, potato, pineapple, and the oriental plant is a huge production in the whole area and the whole world. Then the integration with the genetic engineering also start doing about that. Then the focus for the plant tissue culture and changes to the modern innovation and the 2000, year 2000 present. And then afterwards, the advantage in the molecular biotechnology, genomic biotech, is a use in the tissue culture. That means they are starting is to change the way to do the tissue culture for the technology enhance and then improving in the tissue culture industry. Then afterwards, they are used this MASA system, the TES and automation for the effective micropropagation. Then also the increased use for the tissue culture in the commercial in dangerous species. They have the selected and then the production, and then that skill for the chronic propagation, and then also on for this period for the year afterwards 2000. Okay. Then we look back for this technology for the inventor propagation, and then we have this organ system, somatic, embryogenesis, and then the protoplast culture. And then for this 3 tab, and then also on we had showing about that. Then the biotech is an technology application that they use the biotechnology system, living organism, or the living to make the modified products and then production for the space using the biotech having a great impact on agricultural practice at the all right without the reductions. Then this biotechnology, and then today we had a focus in this tissue culture, molecular genetic, the DNA, genome, and then the gene transfer. Okay, principle of this vector propagation, the aesthetic technique. This one is quite critical because some for the mother plant in the outdoor, maybe it's attack or either it's effect by this disease, and then how to select that is free for carry for the virus disease, and then how to do the sterilization. Then afterwards, use of the explant, and then how to grow, and then to stable, and then reduce the mutation. Okay. Then the third is grow media computation. Is what kind of media is reduce the cost or either can be easy to be used. Okay. Then the third is environment condition. And then inside the lab, normally there will be effect by this light condition, also the temperature, the humidity. And then something that today, some for this indoor, the lab, there we use this called sungo light, for the growing plant culture in a lab, but it's a highly cost. Today more for that is for the fluorescent light change to this LED light. Then the temperature control is also one cost in the lab also. Okay, then is formation for the tissue culture. Plant tissue and organ are go in the vento. Okay. Then this article media is depending kind of the media or this plant where vento. Then we mix come out for the different mutation and then becoming for this application. Okay. Then one is a typical population in the cell and cell development. Then the capital for the cell. Okay. This is basic for this tissue culture concept. How to become another adult plant and then become a new adult plant. And then for the 4 way we can do this selected for this embryo culture, organ culture, or callus culture, or cell culture. And then for this step, we are depending for the kind of the plant we selected, or either it's an ornamental plant or the cash crop or either. And then the horticulture called plant crop, then the ornamental and the floristry species we can use in the commercial application. Then this crop mostly in the tissue culture industry. Okay, the first is a fruit. Normally is tissue culture. We use a pineapple, banana, grape, or strawberry. And then the crop is any kind for the big scale plantation needed. Then the species is something like ginger, turmeric. Then this vanilla also can be used, and then also the ornamental plant. Mostly in the ornamental plant, today a lot is used in the orchid. Okay, and then this global market overview. We look for the potential for the market size and the growth trend, and then this estimation by this CAGR, and then estimation for the next future of another 10 years. The market value for the 0.5 billion USD we increase up to 1.2 billion USD for the crop for this kind of the plant, and then the requirement, and then the purchasing in the power. Then during commercial propagation there will be impact for this industry. Then for the fruit production, mostly in the market today, they have for the material, they have to come out direct for the lab to the market, either from the greenhouse to market, in nursery and a plantation. Okay. Then the economic potential, cost with benefit for tissue culture. Mostly in the tissue culture, the cost is depending kind of the planting material. Give one example. If today we do the tissue culture for orchid, mostly we need in the lab about 20 months, about up to the 2 years. That means in the between these 2 years, we need to injection for capital, everything. That means the cost is higher than the demand. And then the second point, because sometimes for the ornamental plant, they have the seasonal. Maybe example, maybe next year the market purchase lying is a pink color for orchid, but when we produce come out the pink color already, they change away. They want the red color. That means you need to destroy all your planting material, and then this become the cost and the benefit become the comparison. Which one we want to select it? Okay, then the ROI for the nursery and the farmer. Then also the support for government and the private sector. Okay, root is sustainable for the agriculture, reduce the need for the pesticide, and then protection for the free plant. This is more for the farming. They are want to choose the plant for the tissue culture material can become the free for that, and then for the free for pathogen, and then can reduce their cost everything. Then the efficiency for the land use, because in the planting tissue culture material, mostly in the banana planting, if we use a sucker for the banana, the mean from the first harvesting and then the end of the plant, the different time period, maybe it's a different about 3 to 6 months. When you use a tissue culture, you can harvest in the one shot. This is a different way for the industry, and then you can control it. And then the claiming crisis also, you can estimation when you're planting and when you can harvest. Okay. Then the challenges in tissue culture industry. Okay. Okay. Major challenges is a high setup and operation cost. Today, in the lab, we look back for the tissue culture requirement. Mostly is a setup cost, because we need to set up the one tissue culture lab is a small scale. You need to invest about less than 1 million. It's a capacity producer, maybe it's a few million. This is one point. Second point is a labor cost, and then the another point is when you under the production period, then the thermochemical variation, the abnormal cell or either the mutation will happen. You need to restart or either you need to reproduce all the whole batch for this basic, and then this is a major challenge in the tissue culture lab. Then more for the lab in Malaysia. Today in Malaysia, for the small scale up to big scale for the lab, before the COVID, I think whole Malaysia hold the lab more than 120 above. But today I think reduced back to the about 50 or below, because more for the lab at that period shut down. And then they are lost a lot of money, and then there is challenges for that. Okay. Then the conversion risk, because when the period for you're stocking for the stock, and then you cannot take care, you cannot going through, and then contamination will happen. You can need to clear up everything or clear out your whole lab to restart the operation. Then the need of the skilled labor. This is one key point for this industry. Okay, then the planning for strategy for tissue culture company setup, the technical support requirement, training requirement, and then site selection. And then the first for the technical support requirement, because depending for the kind of the plant, the mean if you have the produce different kind of the plant, you need to train your staff for a few different skill. Maybe the orchid production is a different, banana is a different, pineapple is a different, ginger is a different. They mean you must give a lot for the training cost for your staff. Example, if today you only the one mother stock, how you give the training for your staff to do the cutting? Impossible, because the one only one. Then this is facing for this technical this tissue culture industry. Then the second, the training requirement. And then how much the cost you want to input for the issue, the labor cost for the training period. How long? 3 months, half year? This is impossible in the industry, because the cost will impact directly for your output cost also. That means the selling price will be increasing. Okay, we look back for the industry in Malaysia tissue culture. In the past year 2000, at the time period, I sell the banana tissue culture, one plant around 1 ringgit 60 cents. Okay, 1 ringgit 60 cents in the year 2000. Okay, 1 ringgit 60 cents. And the last year, banana become overproduction, tissue culture material. They are throw the price up to 1 ringgit, some for the lab. After 25 years, for the 1 ringgit 60 cents, back to 1 ringgit. But now, last month, they increased back to the 2 ringgit 60 cents. That means we look back, in the 25 years, one banana seeding plant only increased 1 ringgit. How you sustain for your survive, and then how you sustain continuous for your industry. This is for this training requirement, all the things is impact for that. Then site selection, consider also the one key point of the industry also. Okay. Then in the tissue culture industry, we big challenges not only for capital, because we are to know what is happening for the next day or the tomorrow in the tissue culture lab. Is a contamination will happen. We need to restart hold the lab. Example, today you receive a one stock for the sample or the requirement for your client or farmer, and then you produce after half year. Okay, suddenly have the contamination will happen. You need to throw everything, degenerate again, and then this past half year, all the cost you input become zero. Right, burn it. And then this capacity, capital to input, quite difficult to counting how much you want to injection. This is facing by the industry for tissue culture. Okay, then the labor, because the labor training is a kind of the how you train your staff to repeating the job, because only the culture and cutting every day is a repeating, is too boring. And then how you starting for your labor, and then how do you give the incentive either, also the capital for the capacity for the output, because the output depending for the cost for it, and then the one is the impact to the material to the market also. Okay. Then the energy is a high cost, because all the lab you need to sustain, maintain all the light, temperature control, and then also this autoclave to do the sterilization. This is a high cost. Okay. Then another point is you cannot produce a lot for the empty media, waiting for the production for the input, and then you wasted. Okay, then you purchase a big autoclave size, and then you produce maybe one time is a 2,000 glass jar or test tube, but you only use about 100, and other 1,009 you will throw away. The cost will increase for the energy also. Okay, this for the tissue culture changes. Okay. We look back for this strategy for tissue culture company setup. This is a basic capacity for the today, but we changes for the production period. Production period, depending here is showing about 400K up to 800K. Maybe it's higher from this figure. Maybe it's up to 1.5 million. Depending kind of the punting material, how long the period you storage inside your lab, and then the production period, how long you need to come up. Because we look for the tissue culture, most for the tissue culture lab, there is why in Malaysia every lab produce the banana tissue culture? Because first, it's a very short period, about 6 to 9 months you can produce after the market. Okay, this is one point. Then if you produce the orchid tissue culture, you need to 20 months. That means you need to capital to inject between in the 20 months, single income. You continuous inject until you see the planting material come out from the lab, only you can sell to the market. And then if you fail in the media period, maybe it's 3 years. You cannot see the income between in the 3 years. This is the production period. The cost is depending kind of the material or planting material you chooses. And then the lab setup cost also here is showing about 500K up to 1.4 million. It's what kind of the lab requirement you want to set up. You want to class 1, class 2, or class 3. You want this particle counting inside your lab is higher and lower. There will also effect. And then after the lab today, they are facing the third point. Or another point is the nursery, because both for the farmer don't know how to take care the tissue culture material. And then mostly in the Malaysia tissue culture lab, they're facing challenges in that they need to set up their own nursery, take care of the plant until can survive in the farm, or they can sell to the market. Compared with this overseas or Euro country, they are most for the farmer, they have the nursery their own. They only purchase the tissue culture material, go back to do the hardening. But Malaysia, we need to help them to hardening until the plant can sell to the market, can plant. Okay, then the technology innovation is need to change in the automation or bioreactor or article in the media optimized, and then the smart greenhouse. Okay, this okay. Thank you.
Thank you, Mr. Tan, for sharing those valuable insights. Moving forward, we have Mr. Chinagaran as a solution manager, SEED. Today, he will present from farm to future. protecting seed integrity, and enabling trust in Malaysia. Please welcome Mr. Tinagaran.
Hello everyone, good day. Happy Thursday. Yep, looks like I need to do without my slides. All right. First of all, thank you very much for inviting us over here. Good morning, everyone, ladies and gentlemen. My name is Tinagaran. You can call me Tina. And I'm from SIGPA. Next slide, please. Yeah. Yep. Hati-hati benih padi palsu di pasaran. As you know, in Malaysia, agriculture is not only the pillar stone for our economy, but our families. But there is a growing problem within the industry. Fake seeds, counterfeit seeds are going through and seeping into the market. These counterfeit seeds that infiltrate our market are causing disruption not only to the livelihoods of the farmers, but also to the country's food security. Now, what could be the problem that we can be facing? Next, please. Number one is the financial losses for the farmers. Directly, we are talking about $350 million to $500 million of loss in African continent. Now, unfortunately, in Malaysia, there is no study that has been made to find out the impact of exact loss due to counterfeit seeds. However, we cannot be sitting down and not saying that there's no data, right? So we look outwards and we try to learn from what we learn from our global partners. Number two is crop yield reduction. Every time a farmer gets a counterfeit seeds, there is a reduction in their crop up to 50% in certain type of foods. Next, please. And of course, it's the farmer's livelihood is impacted, causing to 30 to 60% loss in their livelihood. Indirectly, this is a problem not only to the farmers, but to all of us. It causes food security and national food security due to shortage, and loses the trust of public towards farmers, the farming industry, as well as the government. Next, please. Yeah. Now, in order for us to be able to understand what is the exact problem, we need to understand why these fake seeds end up in our farmers' hands or in the industry. Number one is the unregulated supply chain. The fact that it's not transparent and opaque is one of the reasons. Number two, lack of seed authentication. How can farmers know or differentiate between fake and genuine seeds? Number three, weak certifications enforcement. Number four, I would say, how can farmer the gap or the awareness gap between farmers and the stakeholders and resistance to new policies. I'm going quite fast in this slide because I'm going to deep dive in every single slide of it. Yeah, so next one please. Yeah. So let's go to problem statement number one. Unregulated supply chains. Today, if you were to buy seeds, there are multiple ways that you can get it. You can import it directly from online, informal channels, manufacturers, even roadside vendors. What does it do? It creates an opaque environment. You would know where seed comes from. Now imagine this. If I were to say each seed or each packaging can have their own passport, not to go to Thailand, not to go to Indonesia, but a passport to be able to give you an identity for the seeds so that you would know where the seed came from. Now, why is it important to have this passport? So that we have some sort of traceability where the seed came from, so that we can make sure that the crops that we are planting are the actual crops that we are planting. Number two, so we're talking about seed passports. I would say